Dear viewers, greetings. In this video, we are going to see about Fourier transform infrared spectroscopy. In short form, it is called as FTIR. This video contains the following contents. First, an introduction about Fourier transform infrared spectroscopy, followed by principle of FTIR spectroscopy, instrumentation of FTIR spectroscopy, working procedure of FTIR spectroscopy, steps in FTIR spectroscopy, applications of FTIR spectroscopy, advantages of FTIR spectroscopy, and finally limitations of FTIR spectroscopy. Fourier transform infrared spectroscopy. FTIR is a spectroscopy technique that uses the mathematic method of Fourier transforms in analyzing the wavelength absorbed by materials. Fourier transform infrared spectroscopy is a method of exploring the physical properties of solids, liquids, and gases. More specifically, FTIR allows the study of the absorptive and emissive properties of materials. The choice of IR wavelength in FTIR method is particularly useful for studying chemical bonds that connect atoms. Principle of FTIR spectroscopy. FTIR spectroscopy is based on the interaction of infrared radiation with matter. The principle relies on the fact that molecules absorb specific frequencies of IR radiation which correspond to the vibrations of the atoms within the molecules. When IR radiation passes through a sample, some of it is absorbed and the rest is transmitted. The absorbed frequencies provide information about the chemical bonds and the molecular structure of the sample. The Fourier transform in FTIR is used to convert the raw data, that is an interferogram which is a function of time or distance, into a spectrum which is also called as a function of frequency via mathematical processing. Instrumentation of FTIR spectroscopy. FTIR spectroscopy contains 5 major parts. They are infrared radiation source, interferometer, sample holder, detector, and computer. The first part of the FTIR spectroscopy is IR source or infrared light source. The infrared light source emits a broad range of infrared radiation, typically from a heated filament. The second part is interferometer. The heart of the FTIR system is interferometer, and this interferometer, usually a Michelson's interferometer, which splits the IR beam and recombines it after traveling different optical paths. This creates an interference pattern. The third part is sample holder. The sample holder can accommodate various samples in various forms like solid, liquid, or gas. The fourth part of the FTIR spectroscopy is detector. Common detectors include a deuterated triglyceride sulfate or a mercury cadmium telluride detector. It detects the interferogram after the IR beam interacts with the sample. The fifth and final part of the FTIR spectroscopy is computer. Computer is used for Fourier transform calculation of the interferogram into an interpretable spectrum. Working mechanism of FTIR spectroscopy. IR source. The system begins with an infrared light source that emits a broad range of infrared radiation. Beam splitter. The infrared light hits the beam splitter, which divides the light into two beams. One beam reflects towards the fixed mirror and the other beam transmitted through the movable mirror. This is the fixed mirror and this is the movable mirror. Third, mirror or fixed mirror. The fixed mirror reflects its beam directly back to the beam splitter. The movable mirror reflects its beam back as well, but since it moves, it creates varying path lights for the beam. This variation in path lights introduces a phase difference between the two beams, leading to an interference pattern where they recombine at a beam splitter. Next, the sample. After the two beams are recombined, the combined beam passes through the sample. The sample absorbs the specific wavelength of the IR radiation depending on its molecular composition. Detector. After passing through the sample, the remaining radiation reaches the detector. The detector measures the interferogram, which is the raw signal of intensity variations due to the interference of the light beams. Next, interferogram. The interferogram is a plot of intensity versus the position of the movable mirror. This shows how the intensity of the IR light changes as the optical path difference varies. As computer or Fourier transform. The interferogram is sent to a computer which applies a Fourier transform to convert the interferogram into a spectrum. Finally, spectrum. The results in the infrared spectrum typically showing intensity versus wavelength. The peak in the spectrum correspond to the different vibrational modes of the molecules in the sample, allowing the identification of its chemical composition. That's all about the working mechanism of FTIR spectroscopy. Steps in FTIR spectroscopy. There are 5 steps. The step 1 is sample preparation. The sample can be prepared as a solid, liquid or gas. Solid samples are often ground with potassium bromide and pressed into pellets, while liquid samples may be analyzed directly or placed between the salt plates. The second step is sample placement. The sample is placed in the IR beam path. Third, interferogram measurement. The interferogram modulates the IR signal, creating an interferogram which is from the raw data. The fourth step is Fourier transform in computer. The computer performs a Fourier transform to convert the interferogram into a spectrum. Finally, the fifth step is spectrum analysis. The resulting IR spectrum is analyzed to identify absorption peaks corresponding to specific molecular vibrations and providing insight into the sample's chemical structure. Applications of FTIR spectroscopy. In organic and inorganic compound identification, FTIR spectroscopy helps to identify functional groups and molecular structures in organic compounds. In polymer analysis, FTIR spectroscopy can be used to study polymers, including degradation, composition, and contamination. In pharmaceuticals, FTIR spectroscopy assists in the quality control and structural analysis of pharmaceutical compounds. In environmental monitoring, the FTIR spectroscopy is used for the detection of pollutants, which are volatile organic compounds, which are present in air and water samples. In food industry, FTIR can detect adulteration and monitor spoilage and evaluate food quality. In biological studies, FTIR spectroscopy analyzes protein, lipid, and carbohydrate structure in biological samples. In forensic analysis, FTIR spectroscopy helps in identifying the unknown substances and residues in forensic investigations. Advantages of FTIR spectroscopy. The first advantage is non-destructive. The samples can be analyzed without causing significant alteration or destruction. The second advantage is speed. FTIR collects all frequencies simultaneously, making it faster than dispersive IR techniques. The third advantage is high sensitivity. FTIR provides detailed information with high sensitivity, especially when using advanced detectors like MCT. The fourth advantage is broader spectral range. FTIR can analyze a wide range of samples in the mid-IR, near-IR, and far-IR regions. The fifth and final advantage is minimal sample preparation. FTIR often requires minimal sample preparation, especially for liquids and gases. Limitations of FTIR spectroscopy. The first limitation is water interference. Water vapor strongly absorbs IR radiation, making it a challenge to analyze water-containing samples. The second limitation is limited quantitative analysis. While good for quantitative or qualitative analysis, FTIR can be less precise for qualitative analysis compared to other techniques like UV visible spectroscopy. The third limitation is sample thickness. Thick or highly concentrated sample may result in distorted spectra due to overabsorption or saturation. The fourth limitation is limited to functional groups. FTIR spectroscopy is primarily effective for detecting functional groups, but it provides limited information about the overall molecular framework. The fifth and final limitation is cost. FTIR spectroscopy instruments, especially those with advanced detectors, can be expensive. Dear viewers, that's all about the Fourier transform infrared spectroscopy or FTIR spectroscopy. Thank you for your support. Thank you.